Hello, my name is Fraser Bye, and I'm a PhD student trying to electrospin a biaxially distensible bilayer membrane for the treatment of cleft palate. The palate forms in the embryo as two palatine shelves that grow down either side of the embryonic tongue, and then at a sharp and well-defined time point they flip up to create the hard palate. It's when these processes fail a cleft palate results. It's an abnormality of the maxilla. This is the bone found at the roof of the mouth. It can either be on both sides or one side, and it can be either accompanied with a cleft lip or without. And it affects somewhere between 1 in 5 to 700 live births worldwide. Here's an example of a cleft palate at maturation, and you can see the extent, especially where the teeth come through, the problems that this will cause. The symptoms of cleft palate are that the child cannot suckle. There's also speech development. The hard surface of the hard palate is required for the formation of many sounds. As your ears are connected to your nose and your mouth through the eustachian tube, it also affects your hearings and also greatly increases the chance of infection developing. And finally, if left untreated, there's a psychological impact on the child, which is considerable. It's currently managed through the use of squeezy bottles. This allows the parent to remove the need for the child to suckle, and the parent can squeeze the bottle and allow the child to feed. Secondly, an acrylic prosthesis known as an obturator can be used. This is fitted to the roof of the mouth and provides a hard surface for a child to push against and allows for suckling and speech development. Each obturator needs to be moulded uniquely to each child, as each cleft palate defect is different. And also, due to the sheer rate of growth of the child, it has to be replaced every 3 to 5 weeks. This is the current treatment timeline for a cleft palate case. As you can see, it's long, protracted, with many stages. From 0 onwards, an obturator may be used. At 3 months, a lip repair is performed. Between 6 and 12 months, a soft palate repair is performed. This is where the hard palate defect is covered in soft tissues. No hard tissues are replaced. Between 1.5 and 5 years, there's ongoing speech assessments and therapy. And from 2 to 15, there's ongoing pre-orthodontics to correct the facial shape, ready for the jaw to bear adult teeth, all the way up to 15, where you get normal orthodontics, where they then rearrange the teeth that do come through. And from 9 years, they receive the only bone graft available, which is to the alveolar ridge, which is this bone at the front of the mouth which bears the adult dentition. Once again, this is not the anything to do with the hard palate defect. This is just the bone at the front of the mouth, so the defect is still there. So to recap the flaws of the current treatments, no hard tissues are replaced. It's only covered with soft tissues. Should you try and place anything in there to allow bone to grow, it must stop the invasion of these soft tissues from growing and filling the void, as they are much faster growing. Secondly, you've got the rate of growth of the child to deal with, which is exponential at this age. Why aren't bone grafts used? Where they have been tried, it's been found that the outcome is worse than not operating at all. This is perhaps due to scar formation and then contraction, which leads to great facial disfigurement. Just to show you how fast a child grows, between the ages of 0 and 5, the rate of growth is exponential. Particularly in the maxilla itself, this more than doubles in volume from the ages of 0 up to 6. So anything that we put in here must be able to cope with this distension. Our ideal treatment timeline would be to use an obturator. Children have a great propensity of putting things in their mouth that they shouldn't, so whatever we put in there needs to be protected. An obturator is the best way to do this. And from about 3 months to a year, we propose to do a complete lip and cleft palate repair, a one-pot fix. And then the orthodontics would still need to be performed, because this wouldn't affect the teeth, but this is a far cut-down treatment regime from the one currently used. In order to do this, we need to create something which will promote the growth of the soft palate. We still need the soft tissues to come in and grow over the defect. It must also allow for the insertion of bony tissue, So we ideally want something which is going to allow ingrowth or the growth of bone inside and result in a new hard palate. Finally, it must prevent invasion of soft tissue in order to give the bone a space to grow and develop. And finally, it must allow for expansion and allow for the rate of growth of the child, so as not to cause facial disfigurement. In order to do this, we intend to create a bilayer membrane. It must be biocompatible and biodegradable. It is unacceptable to have something which requires a follow-up procedure to then remove due to the increased chance of scar formation, facial disfigurement that leads to that from contraction, and also from the increased chance of infection. Obviously, as it's a barrier, it must prevent cells from crossing, but it must also allow the ingress of nutrients to feed whatever's growing in there. If we have a bone there, it must still allow nutrients to get through to this and allow the bone to develop. But as I said, it must be impermeable to cells. And finally, it must deform biaxially. As children don't grow in one dimension, it must stretch in at least 2 dimensions to be able to expand as the child grows. The polymers we are using to investigate this are polylactic acid and polyhydroxybutyrate cohydroxyvalerate. To create these scaffolds, we are using electrospinning, which is based on using a solution of a polymer in a volatile solvent and charged to a high voltage. We typically use 17,000 volts, and then we electrospin onto a rotating drum. There are a number of variables used in electrospinning. Here's just a selection of them. Here is our electrospinning rig. It's quite a simple setup, but we get some quite consistent results out of it and some excellent scaffolds. So, in order to create a bilayer, first we spin a layer of nanofibrous scaffold. The polyhydroxybutyrate cohydroxyvalerate is first spun as a continuous layer. As you can see, it's quite a dense scaffold. Fibre size here is about 800 to 900 nanometers, so we're just in the nanometer range. Once we've coated our mandrel with a dense nanofibrous layer, we can then spin a second layer on top of it. This is of the PLA. As you can see, it's a much more open network of fibres. The fibre diameter is much greater, somewhere between 3 and 5 microns. So, if we spin a nanofibrous layer, followed immediately after by a layer of microfibrous PLA, we end up with a bilayer. And here you can see this is the same amount of material spun for each layer. So we've got the dense PHBV layer at the bottom here, and then we've got this really open PLA network on the top. This is the same mass of polymer. It's both 3 grams of each polymer, but you can just see the density difference between the two. So we're hoping that the PHBV will act as a nice cell barrier, whereas the PLA will act as a very cell-friendly layer and allow the ingress of cells into it. So, we can create a bilayer, but does it stop cell migration? To test this, we've placed the membrane into a cell crown. This is basically an open-bottomed bucket. You can see it here. On one side, we seeded bone-forming cells, which we labelled with a fluorescent dye. And on the opposite side, we placed some fibroblasts, which are known for forming soft tissues, and we fluorescently labelled them with a dye which fluoresces in a different range. We incubated it for 7 days, and then we analysed the results on a fluorescence microscope. The purpose of the open bottom bucket is to prevent cells from sneaking around the outside. The only way that they can go, if they're going to go anywhere, is through. So we want to check to see if they migrate through. So here we're looking at the PLA face. The cells here are a little bit faint, but they are present. So here's a bone-forming HSMP, and there are quite a few of them scattered about. And then you get this faint green fluorescence of the fibroblasts lurking beneath. So you can see them, but they're not quite made it. So here's one poking his head through. So this is great. If you take the sample and then turn it over, this is looking at the PHBV face, and as you can see, it's completely littered with soft tissue forming fibroblasts. And then there's this faint red fluorescence of the HSMPs lurking on the other side, and they haven't migrated through. So this is great. There's been no crossover at 7 days. And just to show you a cross section through this, you can see roughly in blue the scaffold. You've got this red halo of HSMPs on the far side, and this green fluorescence of the fibroblasts on the opposing side. There's no green cells on the red side, no red cells on the green side. The barrier has been effective at 7 days. So, we've made a barrier membrane. It prevents cells from crossing over. The next thing is how to deal with the rate of growth of the child. As I've mentioned before, children don't grow in one dimension. We need something which is going to stretch in 2 dimensions and cope with this rapid growth that we see in children. To do this, we propose to make a corrugated membrane, something which is scrunched up, and that when implanted it can unravel and unfurl as the child grows. So we need a method of creating a crinkly scaffold and then seeing how cells grow on this and how they cope as the scaffold's unfolded. So a method of modelling how the cells cope with a scaffold that undergoes biaxial distension. To do this, we're actually using latex party balloons. They're cheap, autoclavable, and also bio-inert. This makes them great for cell culture. So we get the balloon, we can inflate it to twice the surface area that we require the scaffold to be, spin on a layer of nanofibrous PHBV, deflate the balloon to the required surface area. This crinkles the scaffold by the correct amount to get a doubling in surface area. And then we can electrospin a layer of PLA directly on top to get that nice fluffy layer, great for cells to live in. We can then take the balloon, as I said, spin on the PHBV, spin on the PLA, and then if we autoclave the balloon prior to this, balloons do survive autoclaving, we can then take it and use it for cell culture. So I've basted the balloon in 1 million cells for 20 minutes to try and get some to adhere, and then placed it in a bottle full of media. The balloon is connected via a tube to a syringe pump, and the balloon is filled with PBS. This allows for inflation and deflation of the balloon, thus unfurling the scaffold, giving the cells a workout. And from this we can then see how the cell perform on a scaffold as it changes shape. So we've got the balloon, it's got cells on it, it's coated in scaffold, and it's in a bottle of media. The bottle is placed on a stirrer hot plate to maintain the temperature at 37 degrees, and the balloon is connected via PBS, a PBS tube, to a syringe pump. And from here we inflated the balloon gradually over a week, and we also had a second balloon made up in the same way and placed in an incubator and left as a static culture, so the balloon was not inflated. So we could compare cells on an inflated balloon compared to a statically cultured balloon. To then check to see how the cells responded, took the balloons off after a week, split them in half, and we stained them for MTT. This checked the levels of metabolically active cells. As you can see, we've got good regions of this dense purple dye, so these are all active cells. Okay, there are some patches of white. This is me and my basting technique that needs more practice. Then we also took the opposite side of the balloon and we stained it for cell nuclei using DAPI, and then we also fluorescently labelled it for elastin using immunohistochemistry to see if they produced any more extracellular matrix. If a cell is put under distension, then it should produce more extracellular matrix, and we'd see this as an upregulation of elastin. On the left-hand side, we have a statically cultured balloon. As you can see, there are lots of cell nuclei. There's a little bit of green there, so there's a little bit of elastin, but this is in stark contrast to the exercise balloon, where you can see we've still got plenty of cells there from the blue nuclei, but then we've also got lots of green fibres from elastin. So exercise clearly does make a difference, and it really does upregulate elastin production from cells. So, to summarise, we can create a bilayer membrane of nanofibrous PHBV and microfibrous PLA, and it supports the proliferation of soft tissue forming cells on one side and then bone precursor cells on the opposite side. This membrane can prevent the invasion of cells from crossing from one side to the other, potentially allowing a tissue to be generated inside. Secondly, an expanding scaffold can be created by electrospinning onto a biaxially distensible surface, and then how the cells deal with this surface can be modelled by putting cells onto the balloon and then culturing them while inflating the balloon. Initial results for this are quite promising and shows an upregulation of elastin production if the balloon is inflated while the cells are being cultured onto it. I'd like to thank Professor Sheila McNeil, Tony Ryan and Kayvan, and also Dr. Anthony Bullock for their help and support throughout this project, the University of Sheffield for providing me a home, and the Croto Research Institute and the BBSRC for my funding. Thank you very much for your time.